Hello everyone, I'm Toshiyuki Yokoyama from the University of Tokyo. And today I will talk about semantic variation graphs ontologies for pangenome graphs. And thank you for this opportunity to speak to you today. So this is collaborative work with Pantograph team and ISCB, including some hackathon. And I will talk about introduction and the methodology part. And after that, Simon from University of Tübingen will talk about the result part. Okay, variation graph is a data structure that can encode multiple genomes as graph structure. And there are two genomes with one SNP in the middle of genomes. When we convert this to graph, then graph looks like as follows. Here, common sequences are merged into one node, and variation is represented as branching of graph. And in this graph, a node is a subsequence of a nucleotide sequence, and the edge shows the adjacency between nodes. And path is a list of nodes, which is used to show a haplotype or chromosome of genomes. For visualizing graph genome, our schematized browser shows graph genome on metrics, like multiple sequence alignment. Here, rows are bins and columns are genomes. Group of bins are components. You can see colorful edges between components. We call these links, which represent a nonlinear adjacency caused by genomic rearrangement. This allows you to read off the sequence of any individual by reading the top pangenome sequence and skipping nucleotides that are not present for that individual. We now apply this to visualize SARS-CoV-2 data. And now our Pantograph team succeeded to visualize SARS-CoV-2 data on web browser. If you are interested in this, please don't miss Josiah's talk titled Pantograph Scalable Interactive Graph Genome Visualization in BioViz session. And this is the entire procedure of Pantograph pipeline. Input is a graph genome format. And the OG calculate bins from input graph, and the component segmentation calculate components for each zoom layer. Now we have bins and components and would like to annotate them with existing annotation. This is our proposed pipeline of data flow. VG and the Pantograph data structure can be serialized to RDF using the proposing ontologies. As you know, existing database such as Ensemble or Uniprot has already provided as RDF. Therefore, we'll be able to query against this database with Sparkle and fetch the annotations along with specified subgraph. To enable that, we propose two ontologies. First is variation graph ontology. The important point is how to handle duplication, inversion, or translocation. Due to genomic rearrangement, some nodes can be traced more than twice in a path. To represent this, steps are used by connecting path and node with an index as VG rank. This example shows a simple case, one path with two nodes. This can be easily serialized as the left figure. Second, we propose Pantograph ontology. This is the schematic diagram of Pantograph ontology. This is a little bit complicated, so we'll show how to use this ontology by using a small example. Each zoom level has some components. The order of components can be represented as rank or edge. And also, each component has some bins. The fourth cell, in the schematized matrix, rows are bins and columns are paths. So, intersection point has a reference to bin and path. We call this intersection as cell. Cell has an edge to both bins and regions. If you want to annotate that this cell is inverted, then you can set inversion percent to close to 1. And if you want to say this cell is not occupied by paths, then you can set smaller number of position percent. In addition, in some cases, nonlinear links connect between bins. To represent this, link has departure and arrival to bins. You might wonder how to connect this pantograph ontology to Bio-Ontology. Actually, cell and link have edges to VG path. So this pantograph ontology is able to be inferred from original variation graph. That's the design of pantograph ontology and Bio-Ontology. Then let's move on to the result part.
Hi everyone, I'm Simon Holmus, a PhD student from the Quantitative Policy Center in Tübingen, Germany. Today I will guide you through the second part of this talk, and thank you very much Toshiyuki for this great theoretical overview of the Pangenome ontologies. Now to the practical part. The first time the VG ontology was used for visualization was during the Japan Biohackathon 2019. There we modified the JavaScript code of an existing graph visualization tool called Sequence Tube Map so that we can query a Sparkle endpoint for nodes, paths, and the nucleotide sequences. On the left-hand side, you can see section node query. So the parameters we have taken over from the JavaScript is the starting node ID and the number of nodes. Then we can query all the steps which we need in order to visualize the node. On the right-hand side, you can see the corresponding sequence query. Again, we start from a certain node ID and then collect all the sequences, but the number of nodes we want to have. Here you can see now an example visualization of a query result. We call that matrix tube map, which can be seen as a precursor of our pantograph parser concept. Now let's take a step back and take a look at the traditional semantic web workflows again. What we usually have is we have some data and we create an ontology of the data structure of this data, implement the software so that we can serialize the data into triples, feed these triples into a Sparkle endpoint, and then ask complex questions about the data. However, imagine we have huge amount of biological data like VCF or BAM or PAF. It's very big data, and so it would require an extensive amount of disk space to serialize these data into triples. And the same, of course, holds true for variation graphs. And this problem is tackled by Swatchy, which is used to create a zero extra cost sparkable Pangenome. Swatchy does that by building on OGI, which is a variation graph genome toolkit. It has a very memory efficient C++ data structure implementation, and Swatchy uses the OGI Python bindings to actually wrap an OGI file directly. It introduces IRIs as pointers to the OGI lib-handled graph structures, and in order to relate to linear sequences with other databases like UniProt and the Ensemble EBI-RDF platform, it uses the folder ontology. Now here you can see we are able to directly query a Pangenome graph asking questions via Sparkle without the additional hassle to actually take our data structure and serialize it into Turtle. That's really cool. Of course, Swatchy is also capable of taking our data using the VG ontology and serialize it into triples and feed it into a Sparkle endpoint. That's actually what was done during the Virtual Biohackathon 2020. There, we built a variation graph of a SARS-CoV-2 Pangenome, which is hosted by the public sequence resource. Swatchy was used to translate the OGI graph into a linked Pangenome and also became part of the CWL workflow that is capable of directly serializing the variation graph into Turtle, which is then ready to be fed into a triple store. The ISMB resource currently hosts a triple store of more than 1,300 viral genomes, and together with other resources from INDSC, UniProt, BG, NextProt, OMA, and RIA. So you can ask complex biological questions like, are the active sites for PL1Pro conserved? Now I've shown you some applications of the VG ontology. So let's take a look at the Pantograph ontology. Imagine now we want to use this ontology to actually visualize something. What we need to have there is the information of the bins. We also need the information of the links, and we need the information of the Pangenome sequence. So here on the left-hand side, so you can see the first query, which would give us bins 1 to 5 of zoom level 1. So we select the zoom level, filter by zoom factor. We take the bins and filter by bin rank. Then we also need the cell information. We need the Faldo regions, Faldo begin and end positions, and the Faldo reference, so the path in the graph. And the result of such a query looks something like this. We get the cells, the inversion percent, the position percent, the Faldo begin and end position, and the actual graph, which corresponds to the information in the cell. Now to the link query. Of course, again, we need to query the links which already correspond to bins 1 to 5 of zoom level 1. So we select all the links, we filter again by bin rank, and then we filter by zoom factor. What we get back here are 2 links and the path that participate in that link. And we can also see that the first link starts in bin number 4 and goes to bin number 12. And the second link starts in bin number 17 and goes to bin number 5. What's only been missing so far is the sequence query. That's fairly simple. So all we need to do is select all the sequences of the nodes, concatenate them, and then select from position 1 to 5. Then we have our corresponding Pangenome sequence. Now we could visualize this result, for example, in our Pantograph parser. Yeah, that's all so far. Now let's give you a short summary again. So what we presented you today is the variation graph ontology, which allows you a linear view on things. So it can describe linear sequences in one data structure. And it also has a graphical view, which means that we can relate sequences in a pangenome graph. The Faldo ontology can be used for federated queries and for an exact nucleotide positioning. And Spotgy is capable to serialize our variation graph into the total format. In addition to that, the Pantograph ontology basically is a summarization of the graphical view as zoom levels. The component segmentation ontology branch is also capable of emitting a corresponding total file. And again, the Faldo ontology is used for connecting for the connection with the VT ontology and to generate federated queries. So the current status is that we are one JavaScript library array from running the Pantograph browser on the Pangenome ontology. But there's still lots of work to do, so we are always looking for new members for our Pantograph team. If you're interested, please check out this link. We always need help from grant writers or biologists or software engineers. With this, I want to thank the Pantograph team for their really hard work. And of course, I want to thank you, the audience, for listening. Thank you very much.